The paper I reviewed takes a look at how deep brain stimulation through optogenetics could be a viable therapy for some symptoms of Parkinson's disease. Parkinson's disease is a neurodegenerative disorder that results from the loss of dopaminergic neurons, which leads to abnormal activity in the basal ganglia and altered activity in the subthalamic nucleus. The basal ganglia is largely responsible for motor control, which is why Parkinson's disease can be linked to symptoms like tremors, muscle stiffness, bradykinesia, which is the slowness of moving, and impaired balance and coordination. This study could be relevant in developing a clinical application of optogenetics to alleviate symptoms of Parkinson's disease in humans. So this paper used optogenetics to provide deep brain stimulation to these areas that experience degradation of dopaminergic neurons through viral vector injection to specific parts of the brain with either a channel rhodopsin VCHR1 or CHR2, which are cation channels that hyperpolarize the circuit, or harlodopsin NPHR channels, which is an anion channel that depolarizes the circuit. And yes, I was inspired to continue research on optogenetics from our second paper assignment. So this paper did a within subjects pre-test post-test study design looking at the effects of excitation and inhibition on the subthalamic nucleus regions in adult rats. The rats were also injected with 6-hydroxydopamine in order to exhibit symptoms of Parkinson's, and this injection caused rodents to have specific deficits in contralateral limb use and display rightward rotations of ipsilateral to the lesion. So the results they found looking at inhibition to the subthalamic nucleus of rats with Parkinson's symptoms consisted constant inhibitory deep brain stimulation appeared to not be sufficient in treating symptoms of Parkinson's disease. This was measured through an electrode that can detect spike frequency in the subthalamic nucleus. And the difference in spike frequency before and after opsin activation with blue light was significant, changed from 28 to 5 hertz, but resulted in no measured difference in a behavioral test around percent change in head rotation before and after opsin activation. When looking at optical excitation of the subthalamic nucleus, rats tested with excitatory pulses also showed no behavioral response, indicating that, although not excluding the contributory role, directly generated oscillations within the subthalamic nucleus excitatory neurons are not sufficient to account for therapeutic effects. There was, however, a statistically significant increase in spike frequency, 40 to 82 hertz. It just didn't translate into a strong enough therapeutic results. These tests again resulted in no change with regards to measurement of percent change in head rotation, which is a symptom largely associated with Parkinson's. So to conclude, the study may not have provided a direct answer for therapies related to symptoms of Parkinson's disease, but it highlighted how we are gaining the ability to target specific parts of the brain and either excite or inhibit the circuit. Cortical stimulation could restore balance to Parkinsonian circuitry that is overly devoted to one kind of activity to the exclusion of others, either by disrupting the pathological activity pattern, by promoting throughput of patterns encoding motor behaviors oppositional or compensatory to the lesion pathology, or both. The world of optogenetics is promising for the betterment of Parkinson's symptoms, and this paper is one data point in the right direction for the development of clinical application. Thank you.